Hi guys, and welcome to Biological Sciences for Year 10. This is Activity 11, where we're going to explore in a bit more detail about what is DNA. So in this activity, we're going to look at what are the bases. Now if you can remember back to what we talked about in Activity 10, we understood the idea that basically the DNA is actually made up of single units. Now those single units are made up of 3 molecules. Now that unit is called the nucleotide, which is made up of the phosphate group, joined to a sugar, which is then joined to a base. And remember I said that the bases actually make up the rungs of the ladder. You've got the phosphates on the outside and you've got the bases joining together. What we're going to do in this activity is look at a little bit more detail at those rungs and sort of identify what are the bases. Well, in DNA, there are 4 main bases. Now, they work together in pairs. So one base will always join to another base. Now, if you remember back again to Activity 10, we noticed that we always had on the rungs of the ladder blue going to red and yellow going to green, or green to yellow and red to blue. It's always in that order. You never get things like blue to green or blue to yellow or red to yellow, etc., etc. So what are these bases? Now we are not going to go into the actual chemical makeup of the bases. That will be studied further on if you study biology to a senior level. All you need to be aware of is what are the names of the bases and how do they join together. Okay, so the 4 bases are, we've got thymine. Now thymine will always join to the base adenine. So thymine and adenine go together. And instead of writing the names, we always simplify it using T and A. So T goes to A or A goes to T all the time. The third type of base is guanine, and the 4th is cytosine. So guanine and cytosine, or G and C, go together, or G and C, or C and G, doesn't make any difference. You never get T and C, T and G, A and G, A and C. It's always T to A or G to C. Okay, so if you follow that rule, you can't go wrong. So we've only got 4 bases. So as I say, they basically always join together in those pairs to make the rungs of the ladder. Okay, so let's build the molecule and see how we do it. So basically what we start off with is there's a simple nucleotide which has got the base thymine. So that is going to join with an adenine, always going to join with an adenine. Now you'll see that my adenine nucleotide is upside down, and we'll cross that bridge in a second. So then what will happen is we'll then get another nucleotide which will join to the sugar on the left-hand side, like so. Okay, now that brings that nucleotide of phosphate sugar joins with the base. We just happen to have the base of guanine there. So that guanine will always join with the base cytosine. So you can see we build it up. And what we then get is subsequent nucleotides joining together. Now, it's the order in which those bases join together which is very, very important. The fact that when we read it down either the left-hand side or the right-hand side. And again, we'll talk about that in the next unit. Now, the other important thing that I'd like to talk about here, and this really is just a wee extension of what we're doing with DNA, is to try and identify why is it that it seems that the left-hand side seems to be going up the page, whereas the right-hand side seems to be going down. Well, basically what happens is when we build the DNA molecule, or when the DNA molecule is built, the 2 rungs run anti-parallel. Okay, so one is building one way, the other is building in the opposite direction. So you can see that the sugar has been inverted there. So basically what we get is these 2 strands running anti-parallel to each other. Okay, so it's not particularly complicated, it's just a case of learning. So what do we need to understand? When we put the bases together of the 2 nucleotides, we've got thymine joining to adenine all the time, guanine to cytosine all the time, and the 2 bases just run anti-parallel. Okay, so let's broaden the view of this and see how it all mixes together. So once we've got our complete ladder, once the DNA has been built up, what will then happen is we get our DNA and then we will twist it. Now you may say, why bother twisting? Twisting is very, very important because by twisting the actual molecule, you protect the bases in the inside. And it's the base orientation which is really, really important when we start producing characteristics. If we can protect that base as best as possible, then the result is we are going to get the characteristic coming through without any forms of mutation. Now, mutation will be studied in another activity coming up very shortly. So this twisting basically protects the bases, and we call this twisting a double helix. So let's have a quick look at that and see how it forms. So on the left hand side, what you can see is we've got the bases joined together, and you can go see it goes A to C, C to G, T to A, C to G, A to D, G to C, T to A. You can also see that the left hand side is running anti-parallel to the right hand side. They're swapped over. So once we've got a long strand, at the moment we now need to protect it. So basically what will happen, we will now get that beautiful DNA helix that Watson and Crick basically put forward to us, so we can protect those bases. Now what they actually find is that one complete twist of the DNA helix is made up of 10 individual base pairs joined together. So we've got this twisting, this beautiful twisting of 10 base pairs going from one point right to the next. So as we've gone round 360 degrees, that is made up of 10 base pairs. That twisting protects the bases. Okay, so I hope you found that useful. Again, it gives you that little bit more insight into the DNA and how it's all pulled together. In our next activity, what we're going to look at is how does the bases get converted into a characteristic? How is it that the orientation or the order in which we go, A, C, T, G, how does that get converted into our characteristic? So thanks for watching. Join me again. This really interesting unit on DNA, and I look forward to meeting you again. Thanks for watching.